Recording in progress.
Okay, so this lesson is on sequence analysis. So let's see what the content is. Uh, so in this lesson, at the end of this lesson, I expect you guys to have some knowledge on the types of single sequence analysis, and then the importance of sequence alignment and nucleotide and amino acid sequence analysis. We will also should be able to describe or you should have an idea on various drivers that change sequence DNA sequences, difference between the main forms and alignments, strategies, and their algorithms. Right. So we will be talking about single sequence analysis, sequence alignment, drivers of sequence change in DNA, pairwise alignment, uh, alignment strategies, global and local, and multiple sequence alignment. Right. So those are the things that we'll be talking about in this particular lesson. So, what is sequence analysis? So it is basically the structural and functional examination or analysis of newly determined sequences. It doesn't necessarily have to be a newly sequence or newly determined sequences, but a usual practice is to look at newly sequence or newly determined linear sequences or protein sequences. But as we all know, since um, uh, in most cases, in certain cases, uh, sequences may have been produced for a different reason, but sometimes we can use that same data and examine them, analyze them in a different way, and then we can come up with different kinds of information with respect to those sequences. Right? So that's all what I mentioned about knowledge discovery before the previous lesson. Right? So yeah, you can use the same sequences, not only new sequences but even old sequences, but analyze in a different way and come up with a different view and then generate new information. So one of the fundamental aspects of sequence analysis differences basically. So how are these sequences compared, right? When we are comparing with other sequences, the main comparison comes to the comparison of character patterns, right? So how the nucleotide sequences change. So we look at this by residue to residue correspondence. Right? Basically, position one of sequence one is compared with the same position of sequence two, and we see whether the same nucleotide or whether the same amino acid is being there. Places, the sequences, nucleotides, and amino acids are different. So in order to compare this, the most fundamental step is to align two sequences. What do I mean by aligning? You compare. You make sure you're comparing, you're putting the same residues or same positions of one sequence to another sequence. Right? So basically, you make sure by doing so, you're comparing the same residue of sequence one with that of sequence two. Right? So this is basically making sure that you're comparing apples with apples and oranges with oranges rather than apples with oranges or grapes with oranges and so on. Right? So that's the analogy I took. Right? So we are making sure you're checking the exact positions of two different sequences. So alignment is also the basis of similarity searching, which we conducted before in blast in the blast tool. Right? So it's basically uh, sequence by sequence alignment. Right? We were searching for similarity, but when you are searching for similarity, you all also look for alignment. Right? You look for residue to residue similarity by making sure the sequences are aligned against your query sequence against the database sequence. Right. So let's look at uh, uh, single sequence analysis. Right. So single sequence analysis involves examination and characterization of an individual biological sequence. It can be either a DNA or an RNA sequence or even a protein sequence which consists of amino acids. So the goal of single sequence analysis is to understand the various properties and features of that single sequence without comparing to another sequence. And you look at the number of nucleotides in that sequence, number of adenines, the number of thymines, guanine, cytosines if it's a linear sequence, and then also if it's an amino acid sequence, the different number of different types of amino acids there. And it also involves the identification of the reading frame. If it's a protein coding gene, you need to look at the reading frame, right? So what do I mean by reading frame? Right? So you might end up with a sequence of a protein coding gene, but you don't know whether the first position is the first nucleotide of that gene, or whether it's the second nucleotide, whether the second nucleotide is the first nucleotide of one. Right? So we don't know. If we don't know, we'll have to check the reading frame, right? And then we can also if the reading frame is known, if it's a protein coding gene, if the reading frame is known, we can translate and get the amino acid sequence of that gene. uh, so let's look at the calculation of nucleotide or amino acid composition of a from, the sequence or a protein coding sequence. Uh, so one important thing about looking at the nucleotide composition is that you're of course counting the adenine, thymine, cytosines, and guanine or ATGCs in DNA or if it's RNA. You look at adenine, uracil, cytosine, and guanine. Um, so the important aspect of this is that understanding the GC content. Right? So, for example, the GC content is different in mammals compared to organisms like reptiles and amphibians and other organisms, because mammalian genomes have a higher GC content compared to other organisms. Right? So, what sort of tools can we use to look at the GC content? So one tool is the DNA stats tool in the bioinformatics.org website. Right? So we could just go to that. Mm, uh, right. So I just so I have gone to the bioinformatics bio or do bioinformatics.org website, and I'm looking at. So this is the platform, right? So apart from this particular tool, there are so many other tools also. Um, we can get. Right, I'm going to copy this particular DNA sequence. I copy my paste my copy DNA sequence. So this is DNA sequence, and we just have to just submit, and it will retrieve the number of guanines has been found. So there were 171 nucleotides in the sequence that I submitted. Of that, 56 were guanine, so that's a percentage of 32.75. Adenine 47, again it's a percentage of 27.49. Thymine 29, in a percentage of 16.96. Cytosine 39, with a percentage of 22.81. Right? So there were no other, ambiguous sequences. Mm, So basically, that's it. So it has given the, AT content on the GC 55.56%, adenine and thymine 44.44%. Right. Can you guys hear me? Mm, okay, I think you probably didn't see the output. So this is the output. Sorry, it should be. Yeah, you could see guanines 32%, adenine 27%, and so on. Right. So 171 nucleotides, which 32% were guanine, 27% were adenine, thymine 16%, and cytosine 22%. Right. So that's a very basic stat on DNA or a particular DNA sequence. Then we can also uh, look at protein parameters. And protein parameters. You can get a protein sequence. So to look at the protein parameters, you can just use the protein parameter tool in Expressy website. Uh, Let me share the link. There you go. Right. So this is the protein parameter website. So you just have to copy and paste your protein sequence. So I have done that. And then just click compute parameters, and it will take you right. So protein parameters, number of amino acids 380. His theoretical isoelectric point of PI 7.83, molecular weight is 42,699.62, right? And alanine, 6.8% of the 380 are alanine or 26, arginine 1.8%, right? And so on. So negatively charged amino acids 15, positively charged amino acids 16. Also gives formula, number of atoms, mm, instability index, right? Quite a lot of information from just one sequence of protein or amino acid sequence. And it's broken down into 20, every 10 amino acids, like the 10, 20, 30, and so on. So, uh, uh, in addition, you can also uh, look at the identify the reading frame of a protein coding gene, right? So let's say you have a protein coding gene, but you don't know which place it starts with, right? Which nucleotide, whether it's the first one, second one, or the third one. If you don't know, you can change. You can also check for that, right? So you can do this by using the ORF finder tool in bioinformatics.org. Let's go there again. ORF. Mm, right. So this is the ORF finder tool in the bioinformatics uh, website, bioinformatics.org website. So the options are given here, right? You just ORF, click the ORF finder, and then you take it there. And then this one of the sample sequences. So you could just select submit. So you could say ORF can start with any, right? Unless you have to, if you specify, they'll find a place which that sequence is there, right? So in this particular case, there's nothing like that there. So you could just say any codon, because if you don't know, right? Because you don't know whether your protein starts with this nucleotide, the second one, or the third one. We don't know, right? So codons are repeated every three nucleotides. So we have no idea, right? And there are several options. Right? Search ORFs in the reading frame. One, right? You can also see two and three and one, two, three, and all three options direct. So this says that we can use the sequence in this direction, or else if you specify reverse, it will start reading the checking out the reading frame from this direction. So let's check with that. So and it tells you only return ORFs are at least 30 codons long, right? So if it's less than that, it's don't use. And then use the standard genetic code. So genetic codes, there can be several genetic codes. For mitochondrial genomes, for invertebrates, it's different. Each mitochondria are different. Um, right? So if you know, you have to specify. So let's in this particular case, let's go for standard genetic code. So you have to know this definitely, right? So if this is an example, we'll go for standard and then click submit. So it gives you the uh, the the reading frame number.
Recording in progress.
Okay. Uh, uh, this is basically a very hypothetical nuclear sequence. So it seems like everything is working. Everything seems to be, working, but sequences. Mm, okay. Right. Let's paste this particular sequence. Right. So it's a real gene. We'll try that. Okay. So so translation for first reading frame. Uh, 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 so from base 46 to base 2. Uh, okay. Do you see the results now? So this is the results. Yeah. Uh, so you could see reading frame 3 has a very short protein. Reading frame 2 also has a very short protein. Where there's a you know a protein sequence length around like about 20 amino acids. This one is even shorter. Right? But this one, uh, reading frame number 1, uh, 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 this has a very longer one, and the stop codon here. Right? So so the logic behind this is that you select you select the sequence that has the highest longest reading frame that has the longest amino acid sequence. So in this particular case, reading frame 2 and reading frame 3 are very short, right? So you can't basically have amino acids producing protein this short. Right? But uh, this is the longest one. So reading frame 1, then therefore we select reading frame 1. Okay. Um, right. So let's Back to the presentation. So we can also translate uh, you get the translation from the same method, or you can even use there's a separate tool to translate, again using from Expressi. Uh, uh, so this is the Expressi web tool for translation. Then just Google Translate with Expressi, and then you can just come across this. Uh, uh, so then you can just copy and paste your nucleotide sequence and select the output format. So you just use the verbose this option, compact in no spaces. So we are sure that this is the correct order of the sequence, 5' to 3', and then you select the standard if it's not a mitochondrial gene or other any of these types. So we can just select the standard, right, and then click translate. So so it translates the sequence in three different reading frames: frame one, frame two, frame three. Uh, so uh, open. The so any of these is correct. So, but depending on the presence and absence of stop codons, you can decide which one you should be selecting. So you can also, if you select this option, it will also show the nucleotides against the amino acids. So these are the nucleotides. These are the specific amino acids. So for some nucleotides, codons, there are no amino acids.
Recording in progress.
Right. Okay. Let's try this. This is a intronless gene, and it's a mitochondria. What is it? Mitochondria. Okay. So look at this. This one is very clear. There are no gaps or anything. Uh, just just the amino acid sequence. You just only select contact. It's very clear. Just one of these. So reading frame one is the correct order. So you can't have gaps like this in the middle. Uh, so reading frame two and three are not the appropriate ones. Reading frame one is the correct one. But you can see there's no issues in the middle, no stop codons or anything. Right? So usually stop codons are indicated by these, right? These gaps like such. Right? So you can't have a gap stop codon in the middle of the gene, right? So this one does not have any of those. So then it's very clear this is the correct reading frame and also the actual actual DNA sequence. So let's try. Okay, I yeah, sorry, I haven't shared this. Um, right. Okay, I'll show you expressly. So this is the DNA sequence from a mitochondrial gene. Mitochondrial genes are usually intronless. So I select the compact option and select the gene genetic code divided with mitochondrial, and then I translate with the translation um, in three different reading frames, right? So which one to select is based on the outputs. So you could notice, see these gaps indicate there are stop codons, but then you can't have stop codons in the middle of the protein sequence, right? So reading frame two and three are not the appropriate ones because you have stop codons in the middle. In the middle. So, but this reading frame one does not have any stop codons. So that is the right reading frame, right? And then you can also get the correctly translated protein amino acid sequence for that particular gene. So I only gave the gene sequence from that they produce the uh, amino acid sequence. Right. Or if same thing can be done here. Right? So submitted. Uh, you know, notice that uh, uh, so reading frame two, which is a very star, the star indicates a stop codons. Reading frame three will also have a very short uh, amino acid sequence, right? Stops abruptly uh, with a okay, maybe I didn't. Okay, so I have to okay give the whatever sorry my genetic code. So whatever it whatever mitochondrial is genetic code, and then submit. Right, now we see uh, you can see the results now. Or reading find reading frame. If you use the first reading frame, 954 nucleotides, so it results uh, it ends up translating all the codons in the gene. Right? If you look for reading frame two, right, it stops. Right, reading frame three. Um, uh, yeah, so it stops somewhere in between. Yeah, stop codon. If you go for reading frame three, also that's a very short amino acid sequence. Right, so so that's how you decide. which one is the reading proper reading frame of a particular gene. Right. Stop sharing. Right. Let's get back to the presentation. Right. So I've just basically demonstrated few tools that can be used to determine the amount of the nucleotide frequencies, amino acid frequencies of DNA sequences and protein sequences, and also determine the reading frame and then also translate the sequences into their protein sequences. These are all free, freely available online tools which you can easily use for single sequence analysis. Right. Okay. Now let's move on to sequence analysis. Another aspect of sequence analysis is sequence alignment. Right. So sequence alignment comes when you are comparing multiple sequences, at least one or two sequences together. It involves sequence alignment. Right? So why do we need to align sequences? So one of the biggest issues is that there are differences among two different sequences, even if they're the same gene, right? Or even if they're the same region of the genomic, in the genomes of two different organisms or two different species, there are differences, right? Why are these differences occur? Or why does these differences occur? So we know that DNA sequences and also proteins are all products of evolution. Right? So DNA sequences uh, are the building blocks of life, right? So they macromolecules. So they're linear macromolecules, though, and they determine all the structural and functional basis of life. So uh, uh, they can undergo changes as a result from generation to generation. Right? So one thing is that sequences can generally, when these changes happen, they can accumulate these changes through their genomes. uh, Along with the generation, from the generation to generation, along with time. Right? So let's take this particular example where a particular organism had this particular sequence. Right? Let's call this organism individual, the common ancestor. Right? So that common ancestor had a nucleotide sequence of C, A, A, T, 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 A, T, C, and G. So let's assume about 25 million years later, the there were two populations of rising from this ancestral species that had this particular sequence. Right? However, their sequences were also slightly changed 25 million years later. In this particular place, in this gene sequence, the sixth nucleotide has changed from a T to G in one. In another population, the 10th nucleotide has changed from a G to T, right, in these two populations. Another 25, another 50 million years later, have happened, right? More changes have happened. Another nucleotide has changed in one, where T has been changed to a C. Another change has happened in the other population where C has changed to a T. So, as a result, the populations will genetically diverge over time. Right? They accumulate mutations, and their progenitors will also get these sequences. And and after certain generations, most of the individuals in the population will have these changes in their genome. However, uh, not all regions of the genome will change, right? For example, certain regions of the genome never change, right? However, certain regions they are highly subject to changes. So these are, nucleotides 120 to 180 in the histone H1 protein of humans, chimpanzees, mouse, rat, and cow. You see, even if these organisms evolutionarily have separated long periods ago, you could still see that there are certain regions of the genome. Uh, in this particular case, these are protein coding sequences, which of course directly comes from DNA sequences. Uh, Certain areas there's so much of similarity, certain areas there's so much of differences. So regions where there's no change at all, we call them conservative regions, and regions where there's more likely changes are more likely, and through mutations we call them non-conservative regions. So you can also see the level of conservation of certain regions of the genome through the molecular structures as well. Right? So if you look at the globin genes of vertebrates, you notice they're structurally so similar. Right? If you look at the globin genes of humans, horses, sperm whales, sea turtles, and tuna, you notice the level of similarity, structural similarity. Of course, there could be slight uh, differences in the amino acid sequences. Once they fold into their three-dimensional shape, there is so much similarity, right? Meaning that similarity, I mean, the meaning of structural similarity is that the functionality has been conserved, right? Functionality of these proteins, right? If the globin genes are supposed to transport oxygen and so on, then structurally they have to be the same, despite these organisms are very different. So when we are comparing sequences to alignments, right, we can understand the level of conservation among gene sequences and level of variation among different sites of the same gene. Right? So you could see here, even in this example, certain regions of the sequences are highly conserved. Right? They are identical among so many different species. Right? And then certain sites there's so much that's highlight that's highly changes are very high. Right? High likelihood of being changes. So we call those highly variable regions where changes are very likely, and then there are regions where there's no changes at all. We call those regions highly conserved regions. Right? So we can identify after we align all of the sequences. Right? We can identify all of these differences. uh, So one another thing about this is that if you look at the level of sequence conservation in an alignment, we can understand and we can understand the level of evolution relatedness of different sequences. Right? For example, if sequences are highly similar among different organisms, that is a very good indication of evolutionary relatedness among different species. Right? For example, let's take this particular table where it compares alignments of gene sequences and as well as the resulting protein sequences. Right? So these gene sequences have different names, but then you can align them and you can compare their nucleotide differences. Right? So shark eye control gene, squid eye control gene, fly eyeless gene, flatworm eye control gene, they can all be aligned to the uh, PAX6 gene in the mouse. Right? And when you align them, and you can see there's 85% similarity among the nucleotides in the shark eye control gene to the mouse eye control mouse PAX6 gene. However, there's 78% similarity among squid eye control gene and PAX6 gene. The lowest similarity is flatworm eye control gene and PAX6 gene in the mouse, which is 72%. But interestingly, when you translate the nucleotide sequence to their protein sequence, you find that PAX6 gene protein in the mouse is 100% identical to, amino acids are 100% identical to shark eye control gene, speed eye control gene, fly eyeless gene, and then as well as the flatworm eye control gene. Right? Meaning that the amino acids are 100% the same, comparatively the same to PAX6 gene in the mouse. Which also tells you that these proteins coming from different organisms will still work in the mouse cells as well, right? Why? Because it's the proteins wise they are the same. Although genetically gene sequences are somewhat different, proteins are the same, right? Can anyone tell me why there's there though there are differences in uh, the gene sequences, the proteins are the same. Proteins are identical. Can anyone tell me how is it possible that protein sequences are identical, though the gene sequences are different among the between the PAX6 gene and the others? Use your understanding on molecular biology. Go ahead, guys. What can you say about the genetic code? This has everything to do with the genetic code. If you can just have a very brief idea about the genetic code, you'll notice something where the in a codon, right? The first two nucleotides is the one that matters. Right? The third nucleotide, uh, even if it changes much, it's not going to affect the resulting amino acid. Right? Based on the genetic code, the first two nucleotides are the ones that matters the most. Right? So, if mutations change happens in the third codon position in the different codons in the gene sequences, it's not going to affect on the amino acid sequences. So what can we say about these gene sequences in the sharks? Uh, in the shark, eye control gene, squid eye control gene, fly eyeless gene, flatworm eye control gene, in comparison to PAX6 gene. Right? So most of the changes are most likely occurring in the third nucleotide position. Right? So that is why the amino acid sequence is identical, because changes that happen in these different genes in the shark, squid, and fly, and the flatworm does not go to the protein sequence. Why? Because 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 third position is the most likely position that might have changed. Third nucleotide position is the position that might have changed, resulting to differences in the nucleotide. But accumulating changes in the third codon position in the nucleotide sequences or DNA sequences does not affect the amino acid sequence. So that is why the amino acid sequence are 100% identical, but not the DNA sequences. And you can also understand this by looking at their, their evolution relationships, right? So you notice that mouse and the sharks are vertebrates, so they are the most closely related, and it's also reflected, right? So 85% similarity in the PAX6 gene in the mouse to shark control gene. Right? Squid comes next at 70%, right? Flies are more distantly related and flatworms are more distantly related, only having 72% similarity in the PAC6 gene to flatworm eye control gene. Right. Okay. So, why is it necessary to align sequences? Right? So, for comparative purposes, we need to align sequences. Why is it necessary? Right? So can't you just get your DNA sequences, put them together, and then say, okay, this is an alignment, and you are comparing the actual sites together. Right? Um, Unfortunately, we can't do so. Right? So there are two reasons for that. Right? So look at this particular slide. Right? So you could see there are different sequences. Right? So this is the one you get on the top is before alignment, and once you get on the bottom is after alignment, right? So two things that you can actually notice, right? Or maybe three things. What are the two things? Three things. Tell me one of them. What are the things that you notice here? Do you notice anything? So if you take, one position or one site or one column in this particular data set, you see a lot of dissimilarities among different taxa. Right? These are different taxa. Where you see a lot of differences. Right? But after alignment, you see there are a lot of places the nucleotides are the same. Differences are minimal, but there are more similarities among different sites among different taxa. Right? That's one observation. Right? More similarities after alignment, and you'll also notice the sequences are not of the same length. Right? There are different sizes in the length. Right? So this is basically allows you to see or in not the entire region of the targeted sequence will get sequenced. Right? So from the corners there will be shortening, not because of the evolutionary process, but because of sequencing errors. Right? So sequences, what we get are never of the same length. Right? Another thing is that you'll also notice there are gaps in the middle of the alignment, right? So I'm pointing towards the gaps, right? There are gaps. So those are actually process of evolution where nucleotides can be deleted, or sometimes for other taxa nucleotides can be inserted. So we don't know whether what's happening actually, whether a nucleotide has been deleted or inserted. For others, we have no idea, right? However, in order to compare the exact position of your nucleotides, you'll have to insert gaps to the ones that are shorter, right? Ones that are shorter, right? So once gaps are inserted, you'll be exactly comparing the exact site. Right? So let's say position or site one or site 25 from nucleotide one from sequence one is compared to site 25 from sequence seven. Right? So that is to make sure that you are comparing the exact sites. So alignment process, what it does is that it gets the correct sites of the different sequences in order in the right position, so that you are comparing the right positions or the right sites of the nucleotide alignment. So you call those the correct sites as homologous sites. Right? You are comparing the homologous sites of different nucleotide sequences coming from different taxa. Right. So this is yeah. So so this is before alignment and after alignment. So why is it really necessary? Because you are supposed to, compare apples with apples and oranges with oranges, right? So alignment makes sure that. So therefore, alignments alignment seems to be the most fundamental step in any form of sequence comparison. So how is alignment achieved, right? Alignments are achieved through sequence residue to residue correspondence. Right? You're checking the similarity or matching of each nucleotide to another nucleotide in a different sequence. Right? And then you compare the matching, and then you put up the most appropriate alignment. So if you could remember uh, the algorithm or the strategy behind blast technique that we discussed in the morning, uh, it's basically the same strategy where you compare the residue to residue or nucleotide to nucleotide or amino acid to amino acid level of matching. So again, alignment comes in the basis of similarity searching that we discussed in the morning, and also just basically aligning two sequences for comparison. So one of the major issues of, apart from sequencing errors, another major issue uh, mm, of occurring of comparing between two sequences and the differences that arise, differences that you see between two sequences is because of three types of evolutionary processes. One is substitutions, one is insertions, and the other one is deletions. So let's see what these are. Right. So insertion is the addition of an extra nucleotide to a particular nucleotide, particular sequence, right? So, for example, let's say you start with a nucleotide sequence having GAG, ACT, TAC, right? Just imagine for to the between C and T, between C and T in 1, 2, 3, 4, 5, 6, between the fifth and the sixth nucleotides, right? A new nucleotide has been added, an adenine has been added, right? So that's an insertion. Adenine has been inserted between C and T in the sixth, in the fifth, in the fifth and the sixth position, right? So now the resulting sequence has uh, 10 nucleotides, right? So that's an insertion, right? So afterwards, the sequences will have 10 nucleotides in that nucleotide. And sometimes one nucleotide is being uh, replaced by another one, right? For example, uh, 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 three, four, fifth position, fifth cytosine in the fifth position of the 19 nucleotide sequence is being changed or replaced by an adenine. Right? So number of nucleotides did not change, but only thing is one nucleotide has been replaced by another one. Right? So sometimes the opposite of insertion could happen, where a nucleotide is deleted. Right. So for example, nucleotide C in the which was in the fifth position has been deleted. Right. So GAGAC is now changed to GAGA, right? So what we see is this, right? Uh, Middle this nucleotide in the fifth place has been deleted, C is gone, and you see a G, a G, a G, a T, T, C, right? So, um, so substitutions are quite okay here in the process of evolution because it changes only a nucleotide, it replaces one with another one. But if these changes happens even in the protein coding gene, it can be effective, especially if it happens in the first or the second nucleotide in the codon. Substitutions can affect in the amino acid sequences. But yeah, just imagine if an insertion happens in the middle of a protein coding gene, right? It will entirely change the codons. From that place onwards, the codons can be changed. Right? So as a result, the resulting amino acids will also change, leading to what we call a frame shift mutation. The same can be said again towards deletions, where a nucleotide is deleted, and then from that place onwards, the codons can be changed. Nucleotides in the codons can change, and as a result, that could also lead to a frame shift mutation. uh, Right. So I'll just quickly show you how frame shift mutations can happen. Uh, uh, Give me a second. Right. You see something here. So that's what you see on the top is the nucleotide sequences, nucleotide sequence, right? And you see on the bottom the amino acid sequences, right, on the bottom in different colors. So first codon is saying it could be isoleucine or methionine. Second one is saying it's uh, uh, is third nucleotide codon indicates it's phenylalanine, right, and so on. Right? Sometimes uh, these slash marks indicate it could be either of the two amino acids. But you'll also notice no stop codons. Stop codons are indicated by a black symbol with star mark, right? They are towards the end of the gene, right? So this is the stop codon, this particular gene, right? Right. So just imagine I editing, just enter T here, right? So maybe let's see, where we are, the T here, right? So just remember, the sequence that we had is amino acid sequence that we had is WAIILSGL, right? So just imagine if I enter a T here, right? It changed the entire amino acid sequence, right? And then it also now there are stop codons in the middle also, right? And at the same time, if I delete one, right? Again, the amino acid sequence has changed and it has introduced subcodons in the middle of the gene. Right? So, in case if we right, go back, and if in case if we change the third nucleotide, let's say A with a T, right, it changed that particular amino acid in that particular position, right? It did not change the others. Right? It did not change the others. Right? Sometimes, right? If you had a G, even it does not even change the amino acid sequence, amino acid in that particular place. Right? So the substitutions are less effective, right? And if there are substitutions taking place, it would only affect in that particular position, in that particular position, in that particular amino acid. But insertions or deletions can lead to changes in the entire amino acid sequence, leading to differences in the protein structures, and sometimes it might prematurely stop the protein from producing, because it might introduce stop codons to the middle of the gene. Right. Right. So okay. It's almost 12 now. So we can stop for today, and we will continue with this particular lesson next week also. Right? Right. So do you have any questions regarding what we just discussed? Any questions, guys? Right. If there are no questions, we'll stop for today. Check the LMS. I will upload the reading material and also the video of this particular lecture. Right. Okay. I'll see you next week.
Recording stopped.